Hi, everyone. Welcome to lecture. I'm glad to see that you've made it through your first week. And it means your immune system is working, right? Because if you didn't have an immune system, you wouldn't be here tuning in this Friday, between now and back when we first started talking about the immune system on Monday. Although the truth is, I guess since I can't really see you, I don't know, maybe many of you aren't here, right? Hopefully all of your immune systems are okay and that you are taking part in today's lecture. So a few administrative things I wanted to go over really quick before we get into the lecture today. Here's our class schedule, our semester schedule. I wanted to remind you that you have a couple of assignments coming up. All right. Here we are at the end of week one. Monday, we don't have class because it's the holiday. Right. So the next time I'll see you is next Wednesday. So next Wednesday night, you have your first La Lima quiz due, and it'll be on chapters 1 and 2. And next week on Thursday, you have your first inquisitive homework assignments due, chapters 1 and also chapter 2. So quick reminder that inquisitive, I got that mixed up, right? It's the homeworks are due Wednesday night and the quizzes are due Thursday night. So the inquisitive homework, right? Remember, you need to get onto the inquisitive assignments in La Lima, and you go ahead and you take the interactive inquisitive for each question. You can adjust how many points the question's worth based upon your confidence level when you read the question. You can open up the ebook. They'll have a link in there that actually takes you to where in the ebook the particular topic is. So if you read that, then you can adjust your point value. And you need to gain or earn 800 points in order to complete the assignment. And in doing so, you need to answer at least 10 questions to get there. Right? So the thing about the inquisitives is that it can be time consuming. I think typically it takes students on average between 40 to 75 minutes per assignment, because I think the average number of questions that students answer is over 20. But of course, it goes faster if you take it seriously and you're not just guessing. But it also means, right, that there's no reason why you cannot get 100% for each of your inquisitive homework submissions, right? Because if you just do it and eventually earn your 800%, then you've completed it, right? Okay, so do a few questions per day so they don't pile up. Next Wednesday night, you have the inquisitive chapters 1 and 2 due. So this is one of those weeks a few times during the semester when you have 2 inquisitive homeworks to do, just so that we can get the semester underway, right? Then don't forget you have the quiz, right? That's on La Lima under tests and quizzes, and it's multiple choice, 20 minutes. Do the practice quiz if you haven't already, so that you can actually see what it feels like, what it is, right? And again, right, it'll be on chapters 1 and 2, right? So I will make the first real quiz available after today's lecture, so that those of you who feel comfortable with it and the content can go ahead and take it. Otherwise, you can wait until we go over the rest of chapter 2 next Wednesday, but then you'll just have Wednesday to take the quiz before the due date on Thursday night, right? So you'll just have Wednesday and Thursday to take it. All right, let's continue where we left off last time. We were getting a big overview of the immune system, and we had learned that our immune system is composed of 2 arms, right? The innate arm that has all the stuff that's sort of built into our genes in terms of different types of effector cells and molecules that over evolutionary time have evolved to be able to recognize sort of generic, very common everyday pathogen signals, right, from pathogens that end up infecting our body. Right? And we learned that that's beneficial because it's relatively low cost to maintain, because it's all just sort of pre-programmed innately in your genes, right? And it's very effective. So 99.9% of the pathogens that enter your body today, right, your innate immune system can just take care of. And we don't even think about the fact that every day we experience potentially dozens of individual infections, right? The other side of our immune system is our adaptive side. And so this is when we end up encountering some novel pathogen that might be brand new, your body has never seen it before, and so it doesn't really know how to recognize it well. Or it might be a pathogen that's brand new to the entire human species, right? Like the SARS-CoV-2. As a result, it might be different enough that our innate side of our immune system somehow can't detect it that well, right? And so as a result, that pathogen is allowed to colonize, multiply, and start making you very sick, okay? So we obviously need to have a way to adapt to that, right? Because if you don't come up with a way to mount a defense against that, what's going to happen, right? Just like the piece of moldy bread, right, that doesn't fight back against the bread mold, right? Eventually, all of the resources in your body are compromised, and you will get sicker and sicker, and you will die, right? So the very fact that we get sick but can recover is a testament to our adaptive immune side, learning to recognize that this is in fact a pathogen, right? Identifying different types of signals on it that maybe your innate immune system wasn't trained to recognize, but you have adaptive cells that now can recognize those and use those as the basis of a very powerful attack, right? And importantly, we said part of our adaptive immune system is the ability to remember that identity of that pathogen so that the next time it comes along, you're not back to square one, having to get super sick and potentially dying, right? So I want to point out that the first chapter of the book, it might seem really overwhelming because I don't know why they did this, but in this most recent edition, they've decided to make the first chapter like a huge overview of the entire book, like crammed into one chapter. I mean, I get it. I think it's meant to sort of introduce you and sort of wow you with all these things that you're going to learn about, right? But as a result, it's just so filled with different things, right? Things that in chapter one, they don't go into much detail to actually explain what they're talking about, because the assumption is by the time you get to chapter 10 or 11 or whatever, then you'll talk about, certain things in more detail, right? So as a result, don't be overwhelmed, right? We're still sort of introducing sort of this big range of topics that we're going to talk about. And then once we sort of settle down, we'll then start really looking at each of these things more carefully. So for those of you who have done the chapter one inquisitive, you know what I mean. It seems like a lot of stuff, right? But don't worry about it. It's just an overview, okay? To get you thinking about all the different things that we'll learn about. So the last 3 slides from last time, we didn't get a chance to get to. So I'm going to start off with those. Last time we were talking about our innate system, and I was asking you to think about how our innate system, it's like mall cop, right? Modestly trained, immediate sort of like effector cells that are there and ready to go, and it takes care of most problems, right? Then we have situations where bad clown or exotic pathogen comes along, right? Causes trouble because our innate effectors don't detect him effectively, and bad clown makes us sick, and then we have to have our adaptive immune system. All right, so these slides that we didn't get to last time sort of wrap that up, our discussion. So adaptive training and specialized attack has to do with lymphocytes. So lymphocytes are these cells that are part of our adaptive immune system. And when you hear people talking about B cells or T cells, those are lymphocytes. Right? So the job is that B cells will identify novel pathogens and through the production of antibodies, spread the knowledge of that identified novel threat. Right? T cells then are the molecule or the cells that actually carry out the attack against that pathogen. There are T cells that are helper T cells that help coordinate the attack effort. And there are what we call cytotoxic killer T cells that actually do the killing. Okay. In both of these cases, these lymphocytes begin life as being naive. Naive means they have not ever been exposed or encounter the thing that they actually are able to detect. Okay. Once they have the opportunity to interact with a target like a novel pathogen, right, that this B cell or this T cell happens to know how to recognize, then these lymphocytes become activated. And in the case of B cells, they become what we call plasma cells, and they release antibodies. And in the case of the T cell, they will activate and become an effector T cell, either a helper version or a killer version. So there are different types of receptor proteins involved in this. B cells have what we call immunoglobulins, or IGs, on their surface. That's what these little yellow Y-shaped things are that are attached to the B cell. Okay? When the B cell becomes an activated plasma cell, it produces a detached or soluble version of these same immunoglobulins, and these free-floating immunoglobulins are what we specifically call antibodies, right? So antibodies are soluble, free-floating immunoglobulins. Right? T cells have special receptors known as T cell receptor, or TCR, on their surface. Unlike B cells, T cells never get to the point where they release these TCRs. They always remain on the surface of the T cell. And as we will learn later in the semester, that's very critical for how the T cell functions. Okay? In either case, whether you're talking about immunoglobulins like these free-floating antibodies, or when they're still stuck to the B cell, or you're talking about TCRs, T cell receptors, right? These are very specific shapes, and these shapes determine what types of targeting molecules they can interact with and detect. So a molecule that is detected by an antibody or a T cell receptor, right, that molecule is some kind of three-dimensional shape that matches up with the three-dimensional shape of the immunoglobulin or the T cell receptor. And so that pink dot, right, that target is what we call an antigen. Traditionally, I think you all know antigen, right? You've heard antigen this, antigen that, rapid antigen testing, whatever. So antigen just means the actual three-dimensional molecule that is being targeted by the immune system to be recognized, right, for the adaptive immune system to work. The origin of the name antigen, or the word, is just simply antibody generator, right? So in other words, a three-dimensional molecule that can serve as the basis of your immune system learning how to generate antibodies against it, that is an antigen. Okay. I also mentioned that these different immune cells, like the innate immune cells and the adaptive immune cells, function in different kind of compartments of our body. What I mean by compartments are different tissue types are where immune cells are born and where they develop, right? And in the case of adaptive immune cells, like those lymphocytes, the B cells and T cells, right, there are other tissue types in your body where those lymphocytes actually do their training and their work. Okay, so our bone marrow, our red bone marrow, for example, in the long bones of our body and our pelvis, are where the hematopoietic tissue is found, right? The tissue that gives rise to all of the cells and components, cellular components of our blood and our immune system. So in red are examples of primary tissue, right? The bone marrow in our femur, for example, and the special organ, the thymus, right, which is resting sort of between the lungs. So that's where immune cells are made. And we'll look at this in more depth. Right? Secondary lymphoid tissues in yellow here are specific sites where adaptive immune cells, like the B cells and T cells, have the opportunity to encounter antigen and activate, right? So that they start off as being naive cells, right, that may or may not end up being useful. But if they actually encounter an antigen that they do recognize and bind to, and they activate, that happens in these secondary tissues here, secondary lymphoid tissues. So examples of those are things like the tonsils, lymph nodes, which we'll talk about a lot. We will understand the lymphatic system and its role in adaptive immune response, the spleen, and also specialized tissues that are more spread out. For example, tissues that surround things like our intestines. So gut-associated lymphoid tissues, or GALT. Right? We'll learn about that in due time. Okay. So the last thing I wanted to kind of mention before we get into today's lecture, having to do with this overview of how our adaptive immune system works, is, okay, if the innate side of our system, right, comes pre-programmed and it produces all of these innate cells of our immune system and different types of proteins that over evolutionary history have proven to be useful, right, in being able to defend against like 99.9% of common everyday pathogens, right? That seems like it kind of makes sense, right? You have a set number of cells and they all follow the same game plan. They know what to do when they see pathogens, and they can identify most of the common everyday ones and take care of them. But what about the adaptive side of our immune system? If the whole reason we have an adaptive side is because every so often, inevitably during our life, we encounter pathogens that our innate side doesn't do a good job of recognizing, how does our adaptive side deal with that? Right? What does the whole adaptive nature of it mean? Right? So this is an overview slide that hopefully will at least satisfy your curiosity for now, right? And hopefully won't really totally confuse you, because later when we look at the adaptive system, we'll look at this in more depth. Right? During development and every day of your life, right, you are producing many, many naive B cells, represented here, different colored B cells. These are the Y-shaped immunoglobulins on their surface. What they've done here is they're trying to just really hammer home that each one of these, right, is a different shape. So therefore, each one of these can bind a different three-dimensional shaped antigen. Right? The majority of these, right, probably won't end up being useful for anything. And you're like, what? That's kind of weird. So why do we make them? Right? Remember, what are you trying to achieve here? You are trying to identify something that you have never seen before, and maybe the entire human species has never seen before, right? How do you know what shape is actually useful for binding to that antigen? The bottom line is you don't, right? Your immune system has no clue what to expect or anticipate, and yet it has to be ready to bind to it. So the way we evolved is we produce many, many different shaped immunoglobulins on many, many different naive B cells, right? Each one carrying a particular immunoglobulin. And keep in mind, these cartoons can be very misleading. This makes it look like this one big pink B cell only has one big fat immunoglobulin on it, right? With its own unique little shape. That's not true, right? In reality, the surface of the B cell is covered with thousands upon thousands of the identical immunoglobulin that it alone is carrying, right? The pink immunoglobulin, right? But for simplicity's sake, to illustrate the point, the cartoon shows it this way. So constant activity ever since you were born, producing all of these B cells every single day. Then if you experience an infection, let's say this is a novel pathogen, right? The infection proceeds, your innate system maybe can't detect this pathogen. So as a result, the pathogen is allowed to start spreading and you start getting sick, right? What will happen is that hopefully a subset of those naive B cells that are in your body happen to have immunoglobulins, right, that are reasonably well enough shaped to match the shape of certain features on the pathogen, right? Specifically different antigen molecules that might be on the surface of the pathogen. Right? So as a result, this yellow B cell here gets activated, whereas all of its brother and sister B cells, right, they're the wrong shaped. So they are irrelevant, right? This yellow one will activate, it will clonally expand. And then because it has that unique immunoglobulin on its surface that detects this pathogen, right, that knowledge, right, becomes very valuable. And that knowledge then gets passed on in the form of this B cell communicating with T cells. And the T cells, as I said before, might themselves be helper T cells that then help coordinate a bigger effort to mount this big, powerful defense now, or they might actually be killer T cells, like the SWAT team that actually goes and now eliminates and takes out the pathogen. And we said that takes several days to accomplish because you have to go through these multiple steps of selecting for things, allowing them to build up, and then coordinating, right, this response between different cell types. Okay. All right, so that's kind of the overview of the adaptive immune system. And like I said, hopefully that's not super confusing. We will talk about that in a lot more depth, but I think that gives you enough info to kind of get through chapter 1 and deal with the quiz questions. All right, so let's back up a little bit. So now that we've gotten the overview of both sides of our immune system, let's go ahead and spend a couple lectures just looking at the innate side. Right? As it turns out, maybe this doesn't surprise you, but because our innate immune system is part of how we evolved over hundreds of millions of years, right, ever since the first vertebrates, or even beyond the first vertebrates, the first multicellular complex organisms, right, they had to have some sort of immune system, right? So from that starting point, hundreds of millions of years later, here we are. So it's not surprising then that the way our innate immune system works, right, has many parallels to how the immune systems of other organisms work. And as a result, we know pretty well how our innate system works. There's not a lot of unanswered questions about that. Okay. Just to kind of put that in perspective, I think the book has something like 16 or 17 chapters in it, right? Really only the first 2 chapters deal with innate immunity, right? The whole rest of the book is like adaptive immunity and then trying to piece it together, understanding how it works with innate immunity, right? So that's what we'll do as well. We'll go through the innate immune system in a couple lectures and we'll move on. So the innate immune system has to deal with things immediately. All right, so what we're going to look at today is what we call the immediate response to infection. As a reminder, our first physical defense against pathogens infecting us really are just the physical barriers of our body, membranes, whether you're talking about the tough outer skin or the lining of different types of tracts, like our respiratory tract, GI tract, or reproductive tract. Right? I last time emphasized to you, this is a really important concept of understanding inside versus outside, right? Because even stuff that's going through your respiratory tract into your lungs or food going down through your GI tract are still technically outside of your body in the same way that a car driving through a tunnel, like the polytunnels, right, is passing through the mountain, but is not actually in the mountain, right? And that's a very important distinction. Because our definition of infection kind of only makes sense when you realize that it just has to do with when pathogens somehow cross through those barriers and actually get into our underlying tissues. So let's look at what happens here. Imagine you get a cut on your skin. Right? That moment you get a cut is the moment infection technically starts, right? The moment bacteria on the outside of your skin or on something like the pin or whatever that pokes you actually breaches that protective physical membrane of your epidermis and gets into the underlying tissue, that is the moment infection starts. Fortunately, underneath your outer skin layer in your dermis, you have innate effector cells already patrolling, right? MALCOP is there. Right, in plentiful amounts. And so, for example, macrophages, right? Macrophages are our MALCOP. They are already patrolling in the skin. And so the immediate response is macrophages that happen to be right there, right, come in contact with the bacteria that's coming in. So these rod-shaped, hot dog-shaped bacteria come in. These macrophages then go to work, phagocytosing or eating them and destroying them, right? At the same time, certain receptors on the macrophage are picking up on maybe different types of products of the bacteria that are being released. Right? So that combination of eating the bacteria as well as maybe just picking up on the presence of bacterial signals that are floating around the macrophage, right, trigger the macrophage to go ahead and release little protein messages. These little triangles, right, are small proteins that we call cytokines, right? And the function of the cytokines then is to alert the rest of the immune system that something's going on, right? Like the analogy of Mall Cop, I said that's like Mall Cop who's trained to recognize rainbow hair on the clown, right? And when he sees that, he gets on the walkie-talkie and he says, "Hey, I see a clown here. It's got rainbow hair, and that's what I'm trained to recognize." Right? So that message is the cytokine message going on, and it spreads out. So what are these cytokines doing? Cytokines function to alert the immune system and cause things like inflammation to happen. We will learn about the importance of inflammation and why it's such an important part of our immune response. Right? And importantly, these cytokine signals can attract other immune cells to come and help out. So the recruitment of other monocytes that are floating through the blood, so that those monocytes know that something is going on in the tissue here surrounding the blood vessel, so that they'll squeeze out through the walls of the capillary, right? So this is a capillary in cross section. This is the very thin wall of the capillary. The white space would be the lumen of the capillary in which all the blood is flowing, right? So these monocytes would be recruited by cytokines. They'd squeeze into the solid tissue, and they would finish becoming full-fledged macrophages. Right? Other types of innate immune effector cells like neutrophils would also be recruited, and they would also help contribute. Right. So therefore, there's two sides or two, let's call it stages of our innate immune system. Right? The immediate response are the macrophages and other proteins that happen to be right there, right, to detect immediately and maybe start destroying the bacteria. And then there's the induced response, right, where all the cytokine messages that are released by the macrophages begin to attract or recruit other immune components to help out. Most of the time then, right, this sort of 1-2 punch of your innate immune response is all that you need. Right? So maybe within a few hours, the bacteria have all been eaten up. During that time, your wound is already beginning to kind of heal up, right, and sort of scab over. Right? And so then that's the end of infection, and you didn't even know it. Right? You didn't even know you were infected, and your innate system took care of it. Right? So let's look at this a little closer. So macrophages engulf bacteria via phagocytosis. So this is a really cool image on the left, right? This is an electron micrograph image of a macrophage, this big red cell here. And you can see it is attacking these bacteria, right? And so it is reaching out and grabbing the bacteria, and it's pulling the bacteria in. And you can see these bacteria that are now pressed up against the outside of the macrophage, they're about to get enveloped or taken in. So look at this cartoon here where we can see that, right? So the bacteria get brought to the macrophage, and the macrophage takes it in these what we call phagosomes. Right? And then these vesicles that contain the bacteria end up fusing with other vesicles that contain all kinds of enzymes that would essentially kill and digest and break down the bacteria, right? Thereby neutralizing it. Right? So this poses some interesting kind of challenges. There's only a limited number of bacteria that will happen to be sort of encountered by a macrophage in your skin, right? Or under your skin. It would be much more efficient if the macrophages had a way to really be sort of attracted to the bacteria and grab hold of the bacteria, right? Because the more that the bacteria can be held onto, then the more bacteria a given macrophage can eat, right? So how does the macrophage know where to reach? How does the macrophage know what to grab? Right? Some interesting questions here. Right? I've always loved macrophages. I think macrophages are your like quintessential cliche immune cell, right? Because anytime you see an immune cell pictured, a white blood cell, right? Because remember, macrophages are leukocytes, but there's several different types of leukocytes. But generically, when we see a white blood cell, it's usually this big blob of something that eats something, right? And that's the macrophage. When I was a little boy, I think when I was maybe 10 years old or something like that, I saw this movie on TV. And this was an old movie that was actually made, I think, in the 60s or something. And it was called Fantastic Voyage. And the premise of this movie was, I think it was like the president of the United States or something like that had some kind of like, I don't know, maybe inoperable tumor or something like that, and he was going to die. And so the only way that the doctors figured they could treat him is somehow if they can get in to his body and actually battle the tumor there. All right. And so the way they did this was they got a team of surgeons and they zapped them with this special shrinking ray, all right, that shrunk them down, teeny tiny microscopic, and they shrunk down this kind of like submarine thing for them to like then be in, and then that got injected into the president's body. And the whole movie was about them then going around inside the body and encountering stuff. You know, this is total 60s, right? So that whole idea of like submarines and things was kind of big. But anyhow, so there's this one scene where a big macrophage comes along and it starts attacking the vessel. And on the top of this vessel, there's this like little glass dome where there's like a seat inside. And one of the surgeons is sort of seat belted into this because that's like the observation thing. So this macrophage comes along, right? He gets stuck because his seatbelt won't come undone. And so his fellow crewmates are frantically trying to rescue him, but they can't, right? And then this macrophage ends up like busting through the glass and like eating him, right? And so when I was a kid, man, this was like the most horrifying thing I've ever seen before. But it was awesome, right? So then later, you know, I had my little Star Wars action figures like Luke Skywalker, and I had these like styrofoam, packing peanuts, and I'm like mashing them into the Luke Skywalker. And my mom's just looking at me like, what are you doing? Right? And what are you doing to your toy? And I'm like, Mom, these are macrophages, right? And then I think she just like looked at my dad and she was like, that's your son, right? But anyhow, so macrophages are the quintessential, right, first line of defense in terms of cells that attack things, get into your body, right? And they do so in a wonderfully dramatic manner. Right? But let's think about this. Right? Bacteria have evolved to be very clever in how they can avoid being detected by things like macrophages. The outer coats of many bacteria, especially gram-negative bacteria, have very thick polysaccharide coats on them. So here's an E. coli, right? And if you zoomed in on its surface, what you would see is you have this cell membrane down here, then you have this peptidoglycan layer, and then you have an outer membrane embedded in which are all of these long polysaccharide chains, right? Each one of these little hexagons, for example, right, is an individual sugar molecule, right? And they are attached to these lipids in the membrane. So we call these types of molecules lipopolysaccharides, or LPS. These lipopolysaccharides basically serve as a very thick shag carpet, right? Because three-dimensional features such as proteins and things like that are part of the outer membrane of the E. coli, which could serve as unique three-dimensional shapes that immune cells of your body might be able to pick up on and detect. Those shapes are hidden underneath this LPS layer, right? And so this shag carpeting makes it very hard for things like macrophages to be able to properly attach and grab onto the bacteria. So how do you overcome this challenge? Right? If you have these slippery bacteria, right, with these thick lipopolysaccharide coats, right? How do you have any chance of seeing individual unique proteins inside of them for your immune system to be able to recognize? Well, one thing you can do is you can attach easy to recognize marks to these lipopolysaccharides, right? Sort of marks that your cells have special receptors to easily recognize, and therefore you can make these slippery bacteria much stickier. Okay? So look, here we go. Here's an example. Here I have a beach ball, right? Well, that's a beach ball, but it's actually not a beach ball. It's a very physiologically correct model of a macrophage, right? No, it's not. Okay. And here we have ping pong ball. Right? So this is a slippery bacteria, right? It has this slippery plastic coat. And the bacteria, Oh, this is, by the way, this is a nice example because size-wise, this is pretty much to scale, right? If this is the size of a bacteria, this would be the size of one of your macrophages, right? But the ping pong ball is really slippery, right? It doesn't attach very well and it kind of bounces off. Sorry, I lost it. So how can we make that slippery ping pong ball, right, stick to this macrophage? Well, we can do it through the help of a very particular special group of proteins, right? And these proteins off here, right? You can stick them onto the ping pong balls, right? And now these ping pong balls can be more easily detected. Right? So these proteins is a special family of proteins called Velcro. No, it's not called Velcro, right? These are called complement proteins. And so once you coat the surface of a pathogen with complement proteins, right, they can now stick much easier to the macrophage, right? And the macrophage has a much easier time grabbing onto and more efficiently eating more bacteria. So this requires that what? This requires that the macrophage itself has particular receptors on its surface, right? So in this case, it's the sort of fuzzy felt part of the Velcro. This part that got stuck on the ball is the little teeny microscopic plastic hooks or whatever, right? And so that allows you then to detect the complement proteins, right? So complement proteins coat the pathogen target, and then on the macrophage's surface, right, there would be special complement protein receptors to be able to make it so that the macrophage can then have an easier time binding to the pathogen target, right? Okay. So complement proteins tag pathogens for destruction. Right? So these complement proteins are a system of different soluble proteins that are found in your blood plasma. They're made by your liver. Your liver makes complement proteins, and then your blood is coursing, filled with these complement proteins, right, that then get into your tissues, and they're ready to attach to things like pathogens. So the modes of action of these complement proteins are that they make phagocytosis much more efficient, right, so that the macrophage can eat more bacteria. Also, complement proteins can serve as little signals that attract macrophages. So you can imagine in your tissue where you have a lot of complement protein activity going on, that's probably because there's lots of complement that's coating bacteria, right? So that can serve as a beacon or a sign to attract more macrophages there. And as we will learn, just the coating of complement onto something like a pathogen might already contribute to the disruption of that microbe, right? Leading to, for example, the destruction of the bacteria before something like a macrophage even necessarily has to come along and eat it up. So complement is a system of proteins then, right? It's not just one complement protein. There are many. In fact, there's probably about 30 different types of proteins that are all considered complement proteins. But the most important one is complement component 3, right, or complement C3. Right? So here's an illustration from the book. This blue egg-shaped thing is this sort of super simplified geometric sort of representation of a complement protein C3. Right? There's 3 basic steps as to what happens here. This complement C3 will be floating around in the fluids of your tissue. When it actually comes across something like a pathogen, right, it will be cleaved. So part of it will be cut off. So the C3 then becomes 2 parts, a small part that floats away that we call C3A, and the other part that's left that's called C3B, right? So part 2 then is this C3B is what's going to end up tagging the pathogen. Right? And the part 3 is this will now serve as that easily recognized signal that something like a macrophage that has complement receptor on its surface can now bind to, thereby making this otherwise slippery bacteria suddenly much more attractive and easily bound to the macrophage. This is also the first time I'm going to warn you about the illustrations in the book, okay? Because many of the illustrations of the book are super simplified like this because they're trying to emphasize the concept, but there's a danger in that, right? Because they often overlook or distort things that can be misleading. Number one, for example, complement C3 is not really a blue egg, right? I think you guys know that. It is a folded up protein. Right? And number two, the size scale that's shown here. I mean, this makes it look like one complement C3b is like just as big or even bigger than one bacteria, right? And that's not true at all. Because remember, the C3b itself is just a teeny tiny small protein, right? And the whole outer surface of the bacteria is made of like a bazillion small little proteins, right? So this thing attached to the bacteria, you wouldn't even see it at all if this was drawn to scale, right? But the point is, if the whole surface of the bacterium becomes covered with these individual C3bs and you have a whole lawn or a whole coating of C3b on there, then it is just like the Velcro, right? One individual little plastic hook in the Velcro isn't going to do much. But if you have a whole bunch of them, right, and then you have the other fuzzy felt part of the Velcro pressed up against it, then you get very strong attachment. All right. So how does this C3b part of the complement protein attach to the surface? I mean, what activates the C3 in the first place, right? And how does the C3a part that floats away attract macrophages? Okay. So the complement protein attaches to the surface of a pathogen target using a thioester bond, right? And you're like, oh no, it's OChem. OChem rears its ugly head yet again, right? Yeah, you totally thought that you were done with OChem, right, in your entire life. But no, okay, we do need to talk about a little OChem here, right? So here's a thioester bond, right? So remember, an ester bond is if you have a carbon, double bond to an oxygen, and then you have two different R groups attached to it. So thioester is just a little twist on this, right? Because thio refers to sulfur, so you have a sulfur group in there. Okay. Thioester bonds like this are highly reactive bonds. They love forming covalent bonds with whatever sort of they can, right? So here is the simplified blue egg representation of a complement C3, right? And you'll see that tucked inside of it is this thioester bond, right? Down below is a much more realistic view of what this complement C3 looks like, right? Because again, it is a protein, right? It is a single polypeptide chain. So I think down here, I don't know, maybe this is the end of the, oh no, sorry, over here, right? This is the very end, the very first amino acid of this long polypeptide chain. And if you were to play this little game and follow it around like a maze, right, you'd wind your way all around, all the way around, and eventually you'd reach the C-terminal end, right? The carboxy terminal end and the amine terminal end. So this is where the Bio 275 stuff comes really handy, because those of you who have taken Bio 275, hopefully, yeah, you're like, oh, I remember this kind of stuff, right? For those of you who haven't, and who are right now wide-eyed looking at this going like, I have no idea what any of this means, right? Maybe it's time to find a way to review some cell biology stuff and kind of catch up here, right? I did give you fair warning that Bio 275 is important for this class, okay? Anyhow, so you'll notice that the thioester bond is buried deep within this, right? And it actually comes from the fact that you'll have like two cysteine residues in here that have sulfurs as part of their side chains, right? And therefore they, two cysteines that are next to each other, right, they may interact in such a way to create kind of this little thioester form, right? So the C3 gets cleaved, so part of it falls off, right? And that small part is the C3a that floats away. So that is this part here, this red part here, which is labeled ANA or anaflatoxin. Right? When that floats off, what happens to the rest of the protein? Right? The whole rest of the protein shape has to change to kind of adjust for the fact that part of it has been removed. Right? And as it rearranges, what happens is the part of it that has that thioester bond ends up pointing down, right, over here. And this serves as a nucleophile. Right? And so it undergoes nucleophilic attack on the thioester bond. Right? Because this is floating around in water, you can imagine that almost immediately water interacts with the thioester bond, right, turning it into just a sulfhydryl group and the COOH. And guess what? If that happens, then this C3b is inactive. It's just going to float away and not be useful anymore. Right? But if this cleavage of the C3 happens really close to the surface of a pathogen, there is a chance that the thioester bond will actually form its covalent bond with some molecule that's on the surface of the pathogen, like, for example, one of those lipopolysaccharides, right, that are sticking out. Okay. So there are three pathways that lead to complement C3 being activated. I'm just going to introduce them today. We'll look at these in more depth over the next couple lectures. Right? They're called the alternative pathway, the lectin pathway, and the classical pathway. These all have to do with how different mechanisms take place to eventually lead to the activation, or the scissors cutting the complement C3 so that the C3 can now bind to a target. Right? Today, we're going to start off looking at the alternative pathway. And this has to do with the fact that the pathogen surface, right, really close to it, is a good environment that favors complement C3 to start activating. Right? So the closer the C3 is to the surface of the pathogen, right, the more likely it is it'll activate, and therefore more likely that the C3b will get attached. And this alternative pathway is the first to act. All right. So I'm going to take you through some pretty horrible looking pathways here. Unfortunately, these are some of the more trickier pathways to understand in terms of these protein pathways. And unfortunately, it's at the beginning of the semester. But again, that's just because this is all part of the innate system. So we just have to get through it. But bear with me, and I think you'll be okay. Right? So step one here is, here you have the surface of a pathogen. Here you have a complement C3 that's floating around in the fluid surrounding the pathogen. It gets really close to the surface, right? And what happens is that the C3, right? So this is not C3b yet, right? Because it's floating in water near the surface, it will interact with water, right? And it becomes sort of ready to react. So we call this IC3. This is the first step, right? And the surface of the pathogen, right, is conducive to that because of like the pH and things like that, right? Step two is that this IC3 binds a different complement protein, complement B. That's this yellow thing over here. Okay? And then factor B acts as a zymogen, which basically means it's an enzyme that's going to carry out some function, but in its current shape or form, right, it's inactive. But you'll notice by binding to the complement C3 over here, right, the factor B changes its shape a little bit, and you get this little groove, right? And this little groove is where this next complement protein, factor D, comes in and binds to. Okay? That factor D will bind, and it will cleave this yellow factor B, turning it into a BB and a little BA that floats away. So now this resulting complex we call IC3BB. Right? So what this IC3BB does now is this BB is now active in terms of its enzymatic activity. And what it likes to do is it likes to cleave other C3 proteins. So another C3 protein floats along over here, right? Attaches to the IC3BB, and the yellow part, the BB part, will cleave then this C3 that came along, right, releasing a C3A. And since this is taking place really close to the surface of the pathogen, the C3B, its thioester bond, is exposed and it gets planted there. Right? So that was step 4. Right? So because this complex in step 4 converts C3 into C3b, we call it a C3 convertase complex. Okay. So the next slide continues from this point on, but we'll start with this C3b that's already on the surface, and we're going to move it all the way to the left, right? And here it is. So this process happens again, the exact same process involving factor B, factor D, turning it into a convertase complex, and it cleaving another C3 that floats by, planting that C3b on the surface, right? And once it does that, this convertase complex, after it's given up that newly cleaved C3b, right, it's free to keep going around and around and around, grabbing another C3 and making another C3b. Importantly, each one of these C3bs can also turn into a convertase complex. So once this starts going, very rapidly, you would get this spread or coating of the surface of the bacteria with C3bs, right? You are now putting the Velcro on the surface. Right? And this convertase complex, because it's attached to the surface of the cell, we call this the alternative C3 convertase. Right? So if this is just like the slide before it, what's the difference? The significance is that now you are starting with something that's already on the surface of the cell rather than a C3 that was floating around somewhere else. So therefore, once you get this process started, right, it becomes way more efficient to keep attaching things directly to the surface of the cell. Right? So that's very important to remember. All right. This coating of the surface is what we call opsonization. When you coat the surface of a pathogen target to make it easier to detect, right, you say you are opsonizing that pathogen. Right? So how do we control this? Because obviously this looks like a chain reaction, right? There are certain regulatory proteins that control the deposition of C3b. There are certain factors like this pink protein, properdin or factor B, that stabilize the proper shape of a C3 convertase, thereby making it more efficient for it to carry out its job. So this would increase the opsonization rate of the bacteria. Other factors like factor B or factor I work by cleaving or destroying the C3b that's on the surface, right? So that would slow down the opsonization of the cells. Keep in mind that the thioester bond from the C3 complement, it doesn't discriminate between a pathogen versus like a normal cell in your body, right? So the normal cells of your body might accidentally start getting opsonized, and that would be bad, right? So how do we prevent that? Well, we have certain proteins that are made by our own cells to discourage them from being opsonized. So decay accelerating factor, you can see here, this purple thing removes the yellow BB part from the C3b, right? That would slow things down. Or this membrane cofactor protein, right, over here, right, actually attracts factor I to come and cleave and break up the C3b. Again, slowing things down so that your own healthy cells of your body don't get improperly opsonized and targeted for destruction. Right. This is the last slide for today because I know I'm going over time. So as I explained, right, the process of opsonizing with complement protein greatly increases phagocyte efficiency. So here we have a macrophage. Here we have a bacteria that's being coated with C3b. Again, the size scale is not correct, right? Because the reality would be that you wouldn't see individual C3bs on this bacteria. But the point is, just like the Velcro has two parts, right? Here you've put, one part of the Velcro on here, and then on the macrophage are these complement receptor proteins or receptors, right? CR1 that does the actual recognition, bringing the bacteria in, making it stick a lot better so that the macrophage can more effectively destroy it. Right? So the effectiveness of opsonization through complement proteins is the reason that most pathogen invasions in our body are eliminated right from the start of infection. And within hours, something from a paper cut, right? You're no longer bothered by it because it's already healed up and your innate system has taken care of all the bacteria just through the effects of complement proteins and macrophages. Okay. All right. So that's it for today. If you have any questions in the chat, please feel free to type something in there right now. Okay. So I see some comments here. First chapter is pretty full of content. Yes, it is, right? So that's why I said, don't be overwhelmed with it, because the first chapter is not typical of the other chapters in the book, because it's meant to be sort of this big overview of the entire book. Like I said, I don't know why they did that, because it sort of makes it seem like, wait, the whole textbook could just fit in one chapter, right? So why do you have this big textbook? So don't be intimidated by that, right? Just view it or approach it as this is a big overview of all these many topics that we'll learn about in greater detail, right? And yeah, there is a little animation movie as part of it too, which is pretty cool. And it talks about both the innate and the adaptive side. And I think it probably shows complement proteins also, okay? All right. Yeah, if you would like to unmute yourself, you can ask a question, just as long as you realize that you are being recorded. And if you don't mind that, that's fine, right? So you don't have to type it. So you're welcome to do that. Okay.
Yeah, hi. I just had a question on something you said about slide 16. Is there any way you'd be able to go back?
Yeah, I can do that. Let's go back to slide 16.
I think it was 16. It might have been 17.
This one or the next one?
Actually, I think it was 17.
This one here?
Yeah. Okay. So you see where the C3 that's bonded to the BB, and then it, like that last little step there, does it only create one C3B, or would it make two because two of the C3s are bonded to the BB?
No. So what's happening here is this one. So you can see my cursor moving around, right?
Yeah.
So this is the actual C3 convertase complex that is formed, right? And it's a combination of the original C3 that is now holding onto this activated BB enzyme, right? And so what it does is another C3 comes along, and this shape that forms between the first C3b and this BB is the right shape for another C3 molecule that was floating by to happen to bind to, and that puts it in just the right position for the BB enzyme to do its proteolytic cleavage job, because it is the pair of scissors, right? It now cuts this C3b. So this C3 or this C3 falls apart into C3a and C3b, okay? Then once that falls off and sticks to the surface, this remaining C3 is free to grab another C3 that's floating by.
Okay, I see. So it only cuts one at a time. Like the BB part wouldn't cut both of them. It only cuts the one that comes
right.
and
It
attaches.
only cuts the one because the function of the first C3, right, is merely to create the correct shape being bound to the BB so that another C3 can bind to that shape, and it puts the proteolytic activation site of this enzyme in the right position to cut this second C3. So the first C3 doesn't cut.
Okay, I see. Thank you.
Right. So it just continues to then bind more and start planting on the surface. But that's why I said this isn't super efficient, right? Because what if the C3b that was formed here immediately just binds with water and becomes neutralized and floats away, right? So it's very inefficient. But once you get C3b to successfully stick to the surface, things suddenly get way more efficient. And that's why I said, if you now follow the story of this C3b, we'll move this to the left now in the next slide. Here it is, right? So now it can start forming its own C3b BB complex and continue doing the same process, but because it's starting now being anchored to the surface of the cell, this process is way more efficient to encourage the resulting C3bs that are forming to actually stick right to the surface, rather than being, if this were happening floating around somewhere, most of them would just float away somewhere. Does that make sense?
Yes, thank you so much.
Okay, good. But that's a very good question, right? Because I think many students, you see this pathway first, and then you're like, okay, and then you continues on here, but you're like, wait, this is exactly the same thing. It is exactly the same thing, with the big exception that when on the first one, you're starting with something that's floating around, right? So the efficiency of ending up with a C3b stuck to the surface is far less efficient than if you were to start with something already on the surface and the process continues. It's way more efficient to now get this thing to really coat or opsonize the bacteria. Okay. All right, good. Good question. So thank you for asking that, Megan. Okay. Let's see, other questions. Okay, so Anna, if we were continuously attacked by a certain pathogen and our adaptive immunity responds to it, at a certain point, will the innate response start to incorporate the adaptive immunity response, or it still be part of the adaptive response? Very good question. Okay, the truth is, right, it's not like your innate system gives up or hands things over to the adaptive side once you need to switch on the adaptive side. The innate side keeps working in the background, right? Even though it alone isn't enough to take care of the threat, right? Your adaptive side wakes up right? And you start doing things like getting your lymphocytes activated, making antibodies, but your innate side, all of this stuff, like the complement proteins, macrophages, they're still working in the background, right? So really what ends up beating the infection is a combination of your innate side doing its thing, the adaptive side really doing its thing, right? And an important part of immune memory, and this is why it's really good that you asked this question, is once the adaptive side has really sort of saved the day, right, and helped the innate side take care of things, right, the memory involves next time when the pathogen comes back, right, being able to actually have leftover parts of the adaptive immune system from that experience in the form of antibodies, actually helping out the innate side by doing its thing better. Okay, and we'll learn about that. So that's a very good example of how the two sides actually work together. It's not like the innate side gives up, calls it a day once the adaptive side kicks in. All right. All right, good question. I think it's nice that you thought of that. And we'll see that again, okay, because it's actually a very relevant and very important thing to understand. Okay. All right, I think that's it for the questions. All right, so I hope you guys have a good long weekend. Don't forget to start on your acquisitive homework and also start, thinking about the quiz. You might want to at least review all the chapter 1 stuff so that next week, if you're waiting to take the quiz until Wednesday or Thursday, at least then, you don't have to cram both chapters 1 and 2, all at once, right? All right. All right. Have a good weekend. I'll see you.